Hi everyone, and welcome to this poster presentation on simulating single-cell RNA-seq data using causal generative adversarial networks. My name is Yazdan and I'm a student here at McGill in the Mutz lab. And today we're going to talk about GroundGAN, which is a method we developed. So one of the challenges that GroundGAN tackles concerns the lack of gold standard ground truth when it comes to benchmarking GRN inference algorithms. And we propose using simulation for this task and to generate single-cell data sets for a given GRN. So our goals with GroundGAN were to realistically simulate single-cell data sets about transient state and steady state, but in a way where genes are causally expressed under the expression of the regulating transcription factors. And that is done by letting the user impose a GRN onto the generation process. In addition to that, we also wanted to preserve gene identities, the pseudotime ordering of cells, and to learn the causal nonlinear TF gene dependence, and also other properties of single-cell datasets such as technical noise, biological noise, all of that, but with no user input and only implicit parameterization. So GroundGAN is a causal implicit generative model, and we'll talk about the doors that opens in a second, and as input accepts a GRN and a single-cell dataset. First, we use a single-cell dataset to train a Wasserstein GAN on all TFs and genes. And then we isolate the generator of this first GAN and rename it to a causal controller and use it in the second GAN. So in the second GAN, the causal controller only generates TFs, and those TFs are used as input to another generator called the target generator. And this target generator generates target genes based on TFs and also some noise to introduce stochasticity. And this is where we impose the causal graph or the user-provided GRN. And that is done by letting the target generator's connections follow the connections in the GRN in a way where every single gene will receive as input its own regulating TF. But that wasn't enough to introduce causality, so we also added two auxiliary networks. One of them is called the anti-labeler, the other one is a labeler. And these two networks estimate TF expression given target genes. So that helps us further impose the GRN onto the network. So with GroundGAN, we also show that it generates realistic single-cell datasets. As you can see in this testing plot, there's no evidence of mode collapse. In fact, this is not an issue that we face at all, but it's very common in generative models and across a variety of metrics and datasets. We perform better than the state-of-the-art simulators, as you can see over here, across different distance metrics, maximum mean discrepancy, median integration, least scores, and when we task a random forest to distinguish between cells generated from GroundGAN and real dataset, it performs quite close to random with about 0.54 AURC score. But data replication isn't enough for us to achieve our goals fully. We also wanted to show that GroundGAN causally enforces a GRN. So in this experiment, we infer GRN using GRN boost 2 on real data. And for every single gene, we use half of the inferred edges by GRN boost 2 as positive control and impose those edges onto GroundGAN. The other half we keep and do not impose. And that creates our negative control. And if GroundGAN actually does enforce the GRN, we expect GRN boost 2 and other inference methods to be able to pick up the edges that were imposed. And I mean the ones in the positive control. And that's exactly what happens here. The edges that were in the positive control GRN are now assigned higher scores by GRN inference algorithms and now achieve a higher AUPR on GroundGAN generated data compared to real data. So in this case, it goes up from an AUPRC of 0.29 to 0.77. And the edges that were not imposed in negative control are now assigned lower scores than their scores in real data, so the AUPRC drops down close to random. So we previously talked about how GroundGAN was a causal implicit generative model, and what that allows us to do is to sample from not only observational distributions but also from interventional ones. And that allows us to generate samples under conditions that do not naturally occur in the dataset. And an example of that is in silico experiments, in silico knockout experiments, and various other perturbation experiments. So over here we have this study where we knock out the top 3 differentially expressed TFs of each cell type of the PBMC dataset. And what we see is that cells of the targeted cell type are now dispersed into their neighboring cell types while still being inside the distribution of the single cell dataset. So GroundGAN can be used to generate from interventional distributions. And coming back to our original goal, which was benchmarking GRN inference methods, we benchmarked a number of them, and our results show that GRN boost 2, GNE3, and PIDC perform best across both datasets, and also across different kind of ground truth. And interestingly, Beeline found that many of these methods that do require pseudotime perform quite well on simulated datasets that have very well-defined trajectories, but their performance drops down close to random when run on curated models and real experimental datasets. And our results, ground-GAN benchmarks here, they're all consistent with Beeline's results on real datasets and not with their results on simulated ones. So ground-GAN is also helping close down the gap that exists between real and simulated data benchmarks. So we talked about some of these methods requiring pseudotime for GRN inference. So we also have studies where we show that ground-GAN can be used to generate continuous cell states that are characterized by dynamic processes. And in that study, we show that ground-GAN maintains the pseudotime ordering of cells, and that we can infer pseudotime using PAGA and diffusion pseudotime. And when we do that, we can match branches in ground-GAN trajectories to real ones. And the graph topology and marker gene activation along those trajectories remain consistent with real data. So that was all a quick overview of ground-GAN. And thank you for coming to this presentation. Bye.